All right, so today, guys, we're looking at replication. We'll call it just simply replication because we'll be focused on DNA. Remember, replication is the synthesizing of new DNA. If you watched the last video, went through DNA in general and the history behind it and how all these different scientists led up to finding DNA was the genetic material. Now, we have a couple other players in with replication. Um, we're not going to need to remember them too much. We're going to have to be able to answer these questions once you take your notes. You should be able to answer all these questions. Number one, what is the structure of DNA? From what I've seen, is a lot of people miss that on their pop quiz. The structure of DNA is a double helix. Double helix. That structure was found by Watson and Crick. Number two, you should be able to draw a strand of DNA. Now, when we're talking about drawing a strand of DNA, you need to be able to draw nucleotides and how they are connected. You should remember the types of bonds that form between them. All of these things. Number three, what are the three basic steps of replication? Now, for this one, we did not specifically see this on your notes, but I'm going to talk about it today. So be ready. It'll be the next thing we talk about. Number four, name the 5 enzymes required for replication in eukaryotes. Definitely need to be able to do that. Prokaryotes are a little different. They only require 2, but that's because they go through a much simpler process. They don't have as many steps. Simpler DNA, simpler process. Number 5, you need to be able to tell me what each of those enzymes is responsible for. And then number 6, why do we need to proofread our DNA? I think that question is a very straightforward, meaning why do we need to look back over our DNA? Why would we not want to just submit a rough draft? That's kind of what it's asking. All right. So let's go over a couple things. So let's start with the 3 basic steps of replication. They're pretty straightforward. Now, these 3 steps, you'll notice that they also occur in protein synthesis when we start talking about them. These are just a general idea. They all kind of have the same thing when it goes from process to process. The first step is called initiation. Now, initiation basically means the beginning. So this is where we start. So however replication is going to start, that's called initiation. Now, as you've read through your notes, you've seen that there have been several bubbles that can be formed here, especially in eukaryotes, and that occurs and makes this process go faster. And where we begin, it's called the initiation. It's called the initiation. That leads us into basic step number 2, elongation. This is when we actually add nucleotides. Okay, this is when we actually add nucleotides. Before, we didn't add these nucleotides yet. We had just started the process. For elongation, this is when we add nucleotides. This is when our new DNA grows. If you've read through your notes, you notice that there's a leading and a lagging strand. And these build from the replication fork, and that process is called elongation. Now, there's a certain time when we run out of that DNA, and that process and basic step is termination. That's the end, right? So that's when we stop making new DNA. All right, that's when we run out of things to add. We don't need them anymore. Okay, so if you don't have any more DNA, it's called termination. Now, of course, there's several different factors that go on with each of these. There's initiation factors, termination factors. You don't need to know all that. Just the 3 basic steps: termination, elongation, and termination. That's how we end. Okay? Make sure you hopefully wrote that down, or you just listened to me. Super easy. That's just the basic steps. Now, you also need to be able to figure out this photo that you've gotten on your notes. If you looked at the very end, it showed all the enzymes and where they played during this process. So this is DNA replication, all wrapped up in one picture. So before we get to here, we need to know those 5 enzymes, right? So we need to know the enzymes required. Hopefully, you wrote these down. An easy way for you to remember these, all of these enzymes are super easy in the fact that they end with ASE. That's an easy indicator that they're an enzyme. Remember, enzymes are a specialized protein. They lower activation energy. They allow certain processes to occur. Without these enzymes, DNA replication would not occur. So the first guy we've got coming to the party is an enzyme called helicase. Helicase is one of my favorite simply because if you watch any video, it shows it looking like a helicopter as it flings open and breaks open the bonds that hold these nitrogen bases together. And it's responsible for breaking open the DNA strands. It unwinds the DNA strands. Okay, so if we look back at the picture, we look back at this, we've got helicase right here. There's helicase. It's responsible for unwinding those strands. Helicase. Okay. Now slide on back down. Let's go to the second enzyme, topoisomerase. That enzyme is very important because what it does is it relieves stress and pressure that's going to build from helicase unwinding those strands. So remember, DNA is a double helix, which is a twisted ladder. So as you go to separate the strands, you're going to see there's going to be tension that builds up upstream. Okay. So what happens is we've got an enzyme, let's not use red, use blue, an enzyme that's right here. And its job is to make sure that stress is relieved so that we don't snap our parent DNA molecule. We snap our DNA, that cell's going to die because it can't reproduce, it can't do anything. And so this is topo. I'm going to just write topo. Okay, there's topo. That's topoisomerase. It's there to make sure that the stress is relieved so that we don't snap our parent strands. Okay, now got number 3, third enzyme is going to be primase. Now we've got our strands separated. We've got stress being relieved. So now we've got to actually start building these new strands. Okay. So before we can get started, we've got to have something to kickstart our main enzyme. Okay. We've got one main enzyme that's going to add the nucleotides, but before it can get rolling, it needs to be set up. It's got to have some type of setup. Now, these little dark spots here, you can see how they're a different color. That's because they're a different sugar. And this enzyme that drops these is going to be called primase. Now, we do not have any primase on this picture, okay? They've already come and done their job. They've laid down the particular nucleotides that needs to be there to set up our king enzyme of DNA replication. But without primase, we would not be able to build these molecules. That's why it's very important. Now, a key thing you need to take away from these primers that set it up are RNA primers, meaning they don't have the same sugar as DNA. Remember, the sugar for DNA is deoxyribose. The sugar found in an RNA nucleotide is ribose. So what happens is DNA polymerase can only attach to an RNA primer. So this is Big Daddy. That's the enzyme that's really responsible for building our strands. But it can only attach at spots where it's been primed. So what happens is, this is DNA polymerase, it only attaches to places that will allow it to be attached to those primers. So it waits and waits and waits until finally it finds its primer, and then it starts building those strands. Okay. This, of course, is the most important enzyme to DNA replication, and the simple fact that it builds the new strands. So it actually attaches those nucleotides in. What you'll notice that DNA polymerase can only move in one direction. Okay. Because of this, we get two distinct strands being formed. Okay. We've got one strand that's being made from the origins of replication, that's out here, towards our replication fork. And then we have another strand being made from the fork out towards the origin. Now you can see that these rectangles here, both of these are DNA polymerase. I'll just write DNAP. Got one on each strand here. Now, the strand that's moving from the origin towards the fork and it's made continuously, that's known as the leading strand. And then you have another strand that's moving from the fork towards the origin, that's known as the lagging strand. Now, leading strand made continuously. You can see how this is one complete strand here. It's made very quickly, very easily. Now, the other strand is made in segments. You can see these individual segments here in the lagging strand. This is because DNA polymerase can only move in one direction. It can only move from 3' to 5'. So it has to attach at a certain area that allows it to move in that one direction. Okay. Now, lagging strand is made up of something that's I think it's one of the coolest words. They're called Okazaki fragments. So each of these little fragments has their own little name. They're an Okazaki fragment. They could be 5 nucleotides. They could be 20. Just depends on when the primer was laid. Those are Okazaki fragments. As you would assume, they're named after who discovered them. Now, we can't leave those RNA primers still there. We also can't leave our lagging strand incomplete. So that leads us to our fifth and final enzyme, and that is known as ligase. Okay, so ligase, these little blue guys here, that is ligase. Ligase is responsible for trading out those RNA primers, and it's also responsible for sealing those gaps. So if you look right here, you can see how it is connecting those two strands so that it is a complete strand. Now, those are the 5 enzymes that are responsible for DNA replication. Now, like I said, one of the questions that you have that you should know is, why do we proofread our DNA? Well, again, that's a pretty straightforward answer, with the fact that we wouldn't want to send a rough draft or an incorrect copy to our next set of cells. So what happens is DNA polymerase, it proofreads itself. So it follows those same base pairing rules that you know, A to T, C to G, and let's say it has 3 G's in a row. That means that it should be CCC. That's the complementary strand. Let's say maybe one time it grabs an adenine instead of a cytosine. So what it does is it'll stop, take off that one, and it'll grab the correct nucleotide in order to move on. Now, of course, there are still mistakes, but for the most part, our DNA stays pretty conserved as it moves through our cells. Okay? And that's going to be it for DNA replication. I can talk more about semi-conservative model maybe later, but we'll stop here today. I hope this helps.